Replication Interactive PowerPoint that I showed today. I also wanted to clarify a few things about some of these enzymes we're talking about. Hopefully this will help you as you're studying tonight. So we have one DNA strand, and they're complementary, so that means that A and T are going to pair together, G and C are going to pair together. They complement each other. They're anti-parallel, so this is 3' to 5', while this is 5' to 3'. They're opposite, they flip. So the first thing we have is DNA helicase. That's what's going to unzip the DNA. It's going to break those hydrogen bonds between the DNA bases. So now we've zoomed in a little bit. Then DNA polymerase is going to copy the DNA strand. And it's going to copy in the 5' to 3' direction. Now on here it looks like it's going from 3' to 5'. I can see how that looks like it's going from 3' to 5'. But when I say it's copying from 5' to 3', I'm talking about the new DNA sequence that it's adding. It can only add nucleotides at the 3' end. So it's going to copy from 5' to 3'. So then what you have after it copies is something more like this. This is kind of what you have from that leading strand and the lagging strand. We mentioned those Okazaki fragments, and wherever the DNA polymerase here has to attach to the DNA strand, you're left with a couple of fragments. That's what that ligase is going to join back together. So ligase is going to glue that backbone back together. So as it goes through, it's gluing that backbone back together. And then you have two identical DNA strands. And when we compare that to the original, it's the exact same. They're a match. Now remember, the top half here, this 3' to 5', is what is the template strand for this top strand of DNA. And the bottom strand, the 5' to 3', is the template strand for this set of DNA. So that's why we say it's semi-conservative, because it's not a 100% exact identical from the same material as this one, because it copies new material. So that's why we say that it's semi-conservative. So happy studying, good luck. You will do great on the test tomorrow.